So, most of us think of menopause as this, sudden cliff.
Right, like a switch just flips.
Exactly. We have this mental image where one day the factory just simply runs out of eggs, the lights shut off, and the whole system just powers down. But what if that completely misunderstands the process?
Oh, it definitely does.
Right. What if the real tragedy of ovarian aging isn't this sudden halt, but a slow motion breakdown in the tissue architecture, like a chaotic misfire that is happening while the reproductive cycles are actually still actively running?
Yeah, and that framing, it forces us to entirely rethink fertility and aging.
Yeah.
Because if you were tracking this field, we've spent decades focused almost exclusively on the egg count.
The famous ovarian reserve.
Right, the ovarian reserve. But the research we are decoding today, it shifts the blame away from that really simplistic empty vault theory. Instead, it exposes this massive systemic breakdown in cellular communication.
And the source we have for this is just incredible.
It really is. We are looking at a 2026 paper from Nature Aging, and it's titled Aging Disrupts Spatiotemporal Coordination in the Cycling Murine Ovary.
Marine, meaning mouse, for anyone wondering. And the technology they use to actually see this breakdown is just fascinating to me. It's called Slide-seq.
Yeah, Slide-seq is a game changer.
Because before we dive into exactly what they found going wrong in the aging process, I really want to understand how they mapped this out. I know standard RNA sequencing has been around for a while, to see what genes a single cell is expressing, but this Slide-seq is different, right?
Very different. So, traditionally, to figure out what individual cells are doing, scientists use single-cell RNA sequencing. But to do that, you essentially have to throw the tissue into a chemical blender.
Oh, wow. A blender.
Basically, yeah. You dissolve the physical structure to isolate the individual cells so you can run them through the sequencer.
Which means you get a list of all the materials, but you completely lose the blueprint. I mean, there are bricks and glass, but you have no idea what the house actually looked like or, which room was next to which.
That's a great way to put it. You lose all the spatial context. You don't know who was communicating with whom. And furthermore, the standard method uses these tiny droplets to capture the cells, right?
Well, yeah.
And large, mature oocytes. The egg cells themselves, they often get destroyed because they can be over 70 micrometers wide.
Which is huge for a single cell.
Exactly. They literally don't fit in the machine's droplets.
Yep.
But Slide-seq bypasses that blender problem completely. It's a spatial transcriptomics method.
Meaning it maps the genes out in space?
Right. It allows researchers to map the tissue at a 10-micron resolution without destroying its geography. So in this study, they took 22 naturally cycling mouse ovaries across different ages, and they captured over 610,000 spatial spots.
610,000?
Yeah, to create this incredibly high-resolution, living map of the aging ecosystem.
It's literally like Google Earth for the ovary. You can actually zoom in and see the neighborhoods. So let's zoom in on the main character living in those neighborhoods, right? The oocyte, or the egg cell.
Do it.
Because I think the common perception for most of us is that these eggs are just sitting in a vault for decades in deep freeze, passively waiting to be chosen.
And that couldn't be further from the truth. Oocytes are incredibly busy, even way before ovulation.
Mm-hmm.
They run highly specific, active genetic instructions.
Like software programs?
Exactly like software programs. We call them gene expression programs, or GEPs, as they mature. The paper identifies 3 main programs that are really critical here.
Okay, walk me through them.
Sure. So early on, in the primordial or preantral stages, the egg runs program 2. This is driven by genes like ZP2 and ZP3.
And what are ZP2 and ZP3 actually doing for the egg?
They're building the zona pellucida.
The what?
Zona pellucida. Think of it as a specialized glycoprotein matrix, basically a protective outer shell that the egg weaves around itself.
Oh, like a shield.
Right, but it's essential not just for protection. It's the exact physical structure that the sperm will eventually need to bind to.
Okay, so that's program 2. What happens next?
Later, as the follicle physically grows into what's called the antral stage, it switches to program 4. This uses genes like BMPA15 and these FBOX proteins, specifically FBXW19, 21, and 24.
And their job is what?
They regulate the cell cycle. They are managing the internal growth machinery. And then finally, right before ovulation, it runs program 0.
Program 0, okay.
Yeah, this is the final prep phase. It uses genes like BCL2L10, BTG4, and TCL1 to basically signal that it is absolutely ready for the massive gonadotropin hormone surge that triggers the actual release.
Wow. Okay, so it's almost like an astronaut getting ready for a spacewalk.
Oh, I like that.
Right, because that first program, program 2, is physically putting on the spacesuit. The next program, program 4, is going through all the internal airlock checks, and the final program, program 0, is hitting the button that says, ready for the outer door to open.
That analogy holds up really well, actually, especially when we look at what happens in older ovaries. Because the paper shows us this really profound age-related glitch.
A glitch in the programs?
Yes. The older ovaries don't just have fewer astronauts, The astronauts themselves are malfunctioning.
Oh, man. What kind of malfunctions are we talking about here?
Well, the older oocytes start inappropriately turning on hormone receptors that they shouldn't be using yet. For example, they upregulate R, which is the androgen receptor, and AcR1C18, which is involved in progesterone metabolism.
Why would they do that?
It's likely an act of desperation. The egg is trying to adapt to a scrambled, aging hormonal environment. But the really damaging part is what they turn off.
What do they turn off?
They heavily downregulate a gene called IDE.
Hey, wait, doesn't that stand for insulin-degrading enzyme?
It does.
Why would a microscopic egg cell care about insulin? I usually think of that with, diabetes or diet.
It cares immensely. IDE is absolutely crucial for maintaining metabolic homeostasis inside the cell. When an egg is maturing, it requires very precise energy management.
Okay.
So if you downregulate the exact enzyme that degrades insulin, the insulin signaling goes completely unregulated. The egg gets flooded with metabolic instructions telling it to process energy incorrectly, which leads to massive oxidative stress.
So the cell's internal machinery essentially just burns out.
Exactly, it burns out. And on top of that metabolic chaos, they also downregulate NLRP9B.
And what does NLRP9B do?
It's a gene that is critical for the egg-to-embryo transition.
Oh, wow. Meaning, even if the astronaut manages to get out of the airlock, their life support navigation is completely wiped out. They can't execute the mission of actually becoming an embryo once they're in space.
Precisely. The underlying genetic hardware is compromised before ovulation even happens.
Okay, let me just pause here to make sure I'm holding all these threads together because there's just a lot of incredible biology happening here.
It is a lot, yeah.
So if you're listening, here are the core takeaways so far, just to anchor us. First, we have this new Slide-seq map that lets us see the ovary intact without blending it up. Second, we've learned the eggs are actively building their own protective shells and prepping for launch through these complex genetic programs 2, 4, and 0.
Right.
And third, crucially, aging isn't just a drop in egg numbers, it's an internal glitch where the egg's metabolism goes haywire because it can't manage insulin properly and it loses the instructions for actually becoming an embryo.
That's a perfect summary, which brings up an essential dynamic. The egg isn't doing this alone. It has a massive support system around it.
Right. If the egg is the astronaut, there has to be a mission control. Who are they?
They are called granulosa cells.
Granulosa cells.
Yeah, because a follicle is a multicellular structure. You have the single oocyte in the center, and it's surrounded by thousands of these granulosa cells that feed it, send it signals, and produce hormones.
They're the support crew.
Exactly. But here is a statistic from the field that usually shocks people. Out of all the follicles a mammal is born with, less than 1% ever actually ovulate.
Wait, less than 1%?
Less than 1%.
That sounds like a terrible evolutionary strategy. I mean, biology is usually ruthless about energy conservation. Why on earth would the body waste 99% of its resources building these complex structures just to throw them away?
Because it's not actually a waste. It's a really rigorous quality control mechanism.
Oh, like a competition.
Exactly. The ovary wants to select only the absolute most pristine, genetically sound egg for ovulation each cycle. The other 99% undergo a highly controlled, programmed death process called atresia.
Atresia. Okay, so it's a brutal competition. But how does a follicle actually die? I'm assuming it doesn't just vanish into thin air?
No, it's a very systematic dismantling. And the researchers used the spatial map to track these granulosa cells as they die. They found the cells go through 3 distinct atretic states, and it depends on how far away they are from the egg.
Okay, so to help you, the listener, keep track of these states, think of the acronym MOA. It's like the modus operandi of atresia. MOA. Walk us through how this MOA works.
Good mnemonic. So M is for metabolic reprogramming. This is state 1, driven by genes like CDK8.
Okay.
The cells receive the signal that they aren't the chosen follicle for this cycle, so they start shifting their internal metabolism to prepare for shutdown.
They're packing up their desks.
Right. Then comes O, which is oxidative stress and matrix remodeling. This is state 2, featuring genes like CLU and GPX3. The physical structure of the follicle literally begins to dismantle its own scaffolding.
Tearing down the walls.
Exactly. And finally, A, the apoptotic and immune clearance phase. This is state 3, the terminal state.
Apoptotic meaning cell death.
Yes. The cells are dead, and they use genes like CSH and CD47 to basically send out chemical flares to the immune system saying, "We are done, please come clean us up."
MOA, metabolic, oxidative, apoptotic. So in a young, healthy ovary, a follicle loses the competition, breaks itself down step by step, and the immune system clears the debris. It's a totally clean sweep.
That's how it should work, yes.
But I'm guessing, based on the theme of this paper, in an older ovary, this demolition job doesn't go according to plan.
It fails spectacularly.
Oh, no!
Yeah. In older ovaries, the dying follicles never actually reach that terminal A phase, the apoptotic phase. They stall out halfway through the process.
Why do they stall?
A major culprit identified in this paper is the dysregulation of that CD47 protein I mentioned.
Wait, I've heard of CD47 in cancer research, I think. Isn't that the don't eat me signal?
It is. It's exactly that.
Ah.
In normal biology, healthy cells display CD47 on their surface. It's a chemical flag that tells macrophages, which are basically the immune system's garbage trucks, to leave them alone.
Nothing to see here.
Right. So when a follicle is dying properly, it's supposed to turn that "don't eat me" sign off, allowing the macrophages to sweep in and digest the dead cells.
So in an aging ovary, does the sign get stuck on?
Yes. The dying cells incorrectly keep displaying the CD47 "don't eat me" signal.
Yeah.
So the immune system rolls up to the construction site, sees all this debris, but reads the signs that say, "Healthy tissue, leave us alone."
That is wild.
It's totally confused. The debris is never cleared. The paper describes this as a molecular blurring between healthy and dying follicles.
Molecular blurring. That is such a vivid image. The tissue just becomes this crowded graveyard of half-dead cells. And this has to cause downstream problems, right? I mean, you can't just leave rotting cellular debris in a delicate organ and expect everything to run smoothly.
You absolutely cannot. In fact, the researchers used an algorithm to study this.
Oh, what kind of algorithm?
It's called multi-niche-net. It infers how cells are communicating with each other across the tissue. And when they applied it to this debris field, they found that these stalled, half-dead follicles have highly abnormal metabolic interactions.
Like what?
Well, they specifically highlighted a communication pathway involving a receptor called LRP1. The interaction is APOA4 to LRP1.
Why is LRP1 significant?
Because LRP1 regulates lipid and glucose metabolism. When this pathway goes wrong in the dying follicles, it alters the metabolic environment of the whole ovary. And what's really fascinating for us is that in human genetics, variations in the LRP1 gene are strongly associated with polycystic ovarian syndrome.
PCOS.
Yes, PCOS.
That is incredible. So an algorithm mapping stalled dying cells in mouse ovaries just drew a direct mechanistic line to a major metabolic disorder in humans.
Exactly. It proves that a follicle dying isn't just about clearing physical space. If the process goes wrong, it completely poisons the metabolic well of the whole tissue.
All right, let's do a quick recap so you can digest that before we move to the cycle itself. We know that 99% of follicles undergo this controlled death, transitioning through those MOA phases, metabolic, oxidative, apoptotic.
Right.
But aging ovaries get stuck because the CD47 don't eat me signals confuse the immune system. So the garbage trucks never come, the tissue fills with debris, and it triggers this metabolic chaos linked to things like PCOS. Spot on. So if the ovary is filling up with this confused, half-dead debris, what happens to the timing of the overall system? Because, well, reproduction is all about timing, isn't it?
It is the ultimate biological clock. I mean, in a young, healthy mouse, the physical state of the follicles is exquisitely tied to the hormonal cycle. For a mouse, that estrus cycle runs about every 4 to 5 days.
Okay.
During the estrus phase, which is the exact window when ovulation happens if you look at a young ovary, the next batch of follicles that are waiting in line are physically small. And they have very low expression of gonadotropin hormone receptors, specifically genes called LH-DHER and FBR.
Meaning they have their ears covered, they aren't listening to the hormones circulating in the blood yet.
Precisely. They are synchronized. They are sitting quietly, conserving energy, waiting for the current cycle to finish before they begin their own growth phase. They act as a perfectly coordinated cohort.
So it's almost like a band where Everett is playing together, but if someone is off, it ruins the song.
Actually, it's more like a massive symphony orchestra. In a young ovary, every cell is a musician watching the same conductor, the hormonal cycle.
Oh, I like that.
But the researchers found something in the older mice that completely upends how we view reproductive aging.
What did they find?
Well, these older mice were technically still cycling normally.
Wait, really?
Yeah, if you measure their hormones or track their cycles using standard lab tests, everything appeared totally fine. But under the Slide-seq map, at the cellular level, the timing was completely shattered.
Wait, so the cycle looked normal from the outside, but the cells inside were broken?
Yes, they call it temporal uncoupling.
Temporal uncoupling.
Right. At estrus, when those waiting follicles should have been small and quiet, the aged follicles were already inappropriately large. They were highly mature and aggressively expressing those hormone receptors, the LHE, GR, and FGR, way too early.
So to use the analogy, the musicians were playing the booming crescendo of the second movement while the conductor was still softly guiding the first.
Exactly. It's total cellular chaos.
That is such a vital insight for anyone experiencing perimenopause. You know, you might be going to the doctor, getting your period regularly, your blood work might even look somewhat normal on paper, but at a microscopic level, your tissue is experiencing temporal uncoupling. The cells have literally stopped watching the conductor.
And it shows us that the end of fertility, actual menopause, isn't the beginning of the problem. It is merely the final, visible symptom of a long, slow process where the tissue literally loses its sense of time.
Wow. Let's follow that timeline to its natural conclusion then. Let's say a follicle does manage to release an egg. What happens to the empty shell that's left behind?
It transforms into a structure called a corpus luteum, or CL.
A CL, okay.
This is a temporary but incredibly powerful endocrine gland. Its main job is to pump out massive amounts of progesterone to prepare the uterus, just in case that released egg gets fertilized.
And if no pregnancy occurs?
If no pregnancy occurs, that CL has to be quickly destroyed and cleared away so the tissue can reset for the next cycle.
So we need the garbage trucks again.
Big time. And the paper identified that the corpus luteum goes through stages. The early active stage expresses a gene called PARM1.
PARM1.
Yeah, that's when it's actively making progesterone. But the late dying stage is marked by that enzyme we mentioned earlier, ACR1C18.
Oh, right.
When a CL reaches this ACR1C18 stage, it essentially sends out a massive SOS to the immune system. In a young ovary, a highly organized wave of immune cells rushes in exactly during the estrus phase.
What kind of immune cells?
You have phagocytic macrophages to eat the tissue, antigen-presenting macrophages to coordinate the cleanup, and T cells to regulate the inflammation. They sweep the old CL away cleanly.
Okay, let me guess. In the older ovary, because of all the debris and the broken timing we just talked about, the cleanup crew doesn't arrive on time.
You guessed it. It creates a massive traffic jam in the tissue. The old CLs don't clear out, they just persist and accumulate.
Just piling up.
Exactly. The immune cells do eventually show up, but they are late, arriving in a completely different phase of the cycle, like metastrus. And they are spatially disorganized. They literally wander around the tissue instead of clustering where they are needed.
And if you leave old, degenerating glands sitting around in a tissue, leaking inflammatory signals everywhere...
You get inflammaging.
Inflammaging.
Yeah. This chronic, low-grade, unresolved inflammation is toxic. It causes the fibroblasts in the tissue to panic and start laying down collagen, which leads to fibrosis.
Scar tissue.
Exactly. The ovary literally becomes scarred and stiff. And this physical scarring is the final nail in the coffin for cellular communication.
How so?
Well, in young, healthy ovaries, the tiny, dormant preantral follicles physically clustered together in the tissue.
Like they're huddling.
Yeah, this spatial co-localization is vital because they communicate via paracrine signals.
Yeah.
They basically whisper chemical messages to each other to decide who gets to grow and who stays dormant.
But if the tissue is filled with stiff scar tissue and old, uncleared glands...
The physical space expands and stiffens. The follicles become physically isolated from one another. They can no longer hear each other whispering.
Okay, this is the massive aha moment of the whole deep dive for me. Look at how beautifully and tragically interconnected this is.
There really is a domino effect.
An immune system failure to clean up an old corpus luteum leads to chronic inflammation. That inflammation causes physical scarring in the tissue. That scar tissue physically isolates the new young follicles, preventing them from whispering to each other.
Yes.
And because they can't communicate, they grow at the wrong time, totally uncoupling from the hormonal cycle. The structural breakdown literally causes the timing breakdown.
You've nailed the mechanism completely. A breakdown in physical space leads directly to a breakdown in biological time.
So if you're listening to this and wondering why we are diving so deep into the microscopic geography of mouse ovaries, it's because human perimenopause is notoriously frustrating to navigate.
Oh, absolutely.
The symptoms are erratic, the treatments are often just treating symptoms, and the traditional view is just that you are running out of eggs. But this research validates, at a cellular level, why it feels so chaotic. It proves ovarian aging is a systemic breakdown of cellular timing, immune clearance, and tissue architecture.
And that completely flips how we might think about clinical treatments in the future. Right now, fertility preservation is almost entirely focused on extraction.
Right, getting the eggs out and freezing them before the vault is empty.
Exactly. But if the primary driver of aging is the toxic, disorganized environment of the ovary itself, our interventions might need to change.
We don't just need to save the astronaut, we have to repair mission control.
Exactly. Imagine a future where therapies are designed to anti-age the tissue itself. Treatments that send in signals to clear out that half-dead cellular debris, resolve the inflammaging, dissolve the scar tissue, and physically restore the ovary's natural environment.
It shifts our focus from simply managing a depletion of resources to actively repairing a failure of coordination.
It's a huge paradigm shift.
Which brings us to a final thought for you to ponder. If reproductive decline is ultimately about the cells losing their rhythm and failing to communicate, rather than just disappearing, could we theoretically delay menopause, not by adding anything new to the body, but simply by clearing the static so the existing cells can hear each other again?
That's the million-dollar question.
What if we could just teach the musicians to watch the conductor once more? It's a wildly hopeful way to look at aging. Until next time, keep digging deeper.